I'll tell you a little bit about history. In my opinion, the most beautiful organ in the body, the brain, and this is a three-dimensional reconstruction of an MRI. And what you do not see right here is in the left side of the brain, which is this area right here. Remember, the image is flipped. We have done this right here, in this case, label L for left, and you're looking at yourself at this brain face to face, like this. This is the left side. Underlying this is a very dangerous brain tumor, also known as a glioblastoma multiforme. Over 100 years ago, Santiago Ramon y Cajal, who won the Nobel Prize in Physiology and Medicine because he was able to contribute greatly to the field of neuroscience by looking at the different types of cells that make the brain. He said that we had a harsh decree that everything may die and nothing may be regenerated in the adult central nervous system. He left the possibility open for pathological diseases. He was a pathologist by training and became one of the great neuroscientists in the world. Over the next 30 to 40 minutes, I will take you over some of the history of the controversy of the adult neurogenesis, the difference between the human versus the rodent stem cell niche, more specifically the subventricular zone. I will take you over whether or not there are stem cells that are found in the human brain and whether or not there are stem cells found in human cancer. The question that still remains is whether or not a neural stem cell or a stem cell may give rise to a brain tumor stem cell, and that is work in progress, and I will elaborate on a little bit of that. What about this issue of the controversy of adult neurogenesis? Well, it turns out that dating back to the early 1900s, even Santiago Ramon y Cajal knew that we had limitations in technology. He said once that none of the methods used by these investigators are capable of distinguishing absolutely a multiplying neuroglia cell from a small mitotic neuron. So the question is whether or not there were new neurons being formed in the adult mammalian brain. By the 1960s, Joseph Allman, doing a study with thymidine autoradiographic evidence, he was able to find new neurons in the adult rat and cat. But it would take a few years later, by the 1970s, when Michael Kaplan, he combined thymidine labeling and electron microscopy. This is the early resurgence and the early utilization of electron microscopy that confirmed Altman's claims by showing mitotic neuronal precursors lining the lateral ventricle. But people still were very skeptical. It was kind of hard to understand and to believe that indeed new neurons are being produced in the lateral ventricles of the mammalian brain. By the 1980s, Notenbaum and colleagues out of New York were, by doing some really eloquent studies in the avian brain, they were able, through a series of experiments, show several things. The production of new cells with thymidine labeling. They were able to show that these new cells were not only neurons, but they were also receiving synapses. And most importantly, these neurons that were receiving synapses responded to sound with action potential. Yet, people were very skeptical about the issue of neurogenesis and the avian brain because they said that it was not the mammalian brain. So by the 1990s, work in the rodent, out of several laboratories around the world, they began to unravel and teach us more about this issue of the controversy of neurogenesis. By 1997, a group of investigators led by Dr. Arturo Alvarez Buya, also out of New York and subsequently out of California, was able to elucidate the organization and cytoarchitecture of the rodent subventricular zone. By 1999, a very important discovery that was also controversial, several groups believing that were probably ependymal cells, the ones responsible for neurons, and Arturo and his group believing that it was actually astrocytes, was able to show that an adult neural stem cell comes actually from astrocytes in the rodent brain. What was not known at this point was whether or not this was also true for the human brain. It was even less known the difference in this stem cell niche, the subventricular zone, between the human and the rodent. And that's the next portion of this discussion. Several years later, by the year 2003, 2004, as Arturo and his group and myself involved in some of these studies, we began to unravel some of the mysteries of the organization of the human subventricular zone. We began to look at markers of migration, in this case PSA and CAM, markers of proliferation, Ki67, markers of neuronal precursors, TUJ1. And we look at several parts of the brain. This is anterior horn, the body of the ventricle, the other parts of the ventricle, as you can see, all the way to the temporal horn right here on the bottom. And what you can see, actually, not only the organization was different, there was evidence of proliferation, migration, but also we found evidence of some neurons struggling into this area of the subventricular zone going up and down. So after many years of looking at the organization of the rodent subventricular zone done by many groups around the United States and the world, we began to look at the organization of the human subventricular zone. And you can actually see here in red is GFAP and in blue is DAPI. For the rodent subventricular zone, you can actually see GFAP in green, double cortin in red, and DAPI in blue. And then there's a cartoon illustration between the human subventricular zone and the rodent subventricular zone. The rodent subventricular zone, not only is the brain a little bit more lysencephalic, is called, that means that it has less groups as compared to the human brain, but in the area of the subventricular zone, that is the lateral wall of the ventricle right here, this is an amplification, you can actually see a group of cells just coming out, also known as the rostral migratory stream, and these astrocytes are completely welded against the ependymal zone. Yet, in the human brain, there's the ependymal zone right here, which is called layer 1. Then layer 2 is a hypocellular gap. Then layer 3 is a ribbon of astrocyte. And then there is a transition to the parenchyma. Striking differences between the organization of the human subventricular zone and the rodent subventricular zone. So we got interested in our laboratory to further elucidate the organization of the human subventricular zone. We went on and do these very similar studies in the fetal human brain. And indeed, it appears to be that the lateral ventricle may be connected also to the rostral migratory stream all the way down against the olfactory bulb. This is a so-called sagittal section along this region that is slightly oblique right here and a little bit inclined, as you can see right here. And we were able to study different regions of this part of the brain, trying to understand how is the fetal brain and the human brain organized as compared to the rodent brain. Is there a population of cells that are migrating? Well, it turns out that we found some evidence that indeed there are some cells that appear to be migrating outside of this region. Not necessarily the same way that they appear to be migrating in the rodent brain, from the ventricle down to the olfactory bulb. But indeed, some of these cells appear to be migrating in groups, and they are surrounded by these glial cells. This is a confocal microscopy of such a region in the human fetal brain. Unfortunately, our group have not been able to find evidence of a similar type of migration in the human brain per se, in the human adult brain. But other groups around the world claim that this does indeed exist. So the question is, if migration exists in the human brain, could we learn something about that migration that will help us understand why brain tumors are so migratory? This is an image that I will show you of a small piece of human fetal brain that is put on matrigel, in which you're going to see evidence of migrating cells surrounded by glial sheets. And this is the picture right here. This is the movie. You can actually see. This is from our own laboratory. Cells migrating. This is human fetal brain. They're migrating out, and they are surrounded. And we've done all these immunostainings to be able to see that indeed the human fetal brain may have some similarities in regards to migration as compared to the rodent brain. We have done similar studies, as you can imagine, with human brain tumors, and we are in the process of trying to unravel some of the mysteries that human brain tumors use for migration. I'll take you. So we have gone over the controversy of adult neurogenesis. I think it's clear that the mammalian brain does have the ability to generate some neurons, at least in the rodent brain, and similar evidence may actually be coming more clear in the adult human brain. We know that the stem cell niche, in this case the subventricular zone, is different between the human and the rodent. The question is whether or not there are stem cells in the human brain and whether or not there are stem cells in human cancer. So, could it be that there are adult human neurostem cells? By the 1990s, Steve Goldman and his colleagues, they did several studies and published a paper about the finding adult human neurogenesis from temporal horn subventricular zone in vitro. This was from temporal horn surgeries in which they did temporal lobe resections and were able to culture this tissue and create neurons. For the first time, showing that indeed, at least in the petri dish, this is possible. By the 1990s, Gage and his colleagues showed a study by looking at brains post-mortem, although they call it in vivo, but post-mortem, in which they show neurogenesis in the adult human hippocampus. And I am right side of the hippocampus right now. I'm showing one specific picture, and where you can actually see right here in this particular cell right here that is marked with two markers. Red is nu-N, as you can see right here, and green is BRDU. So this appears to be the formation of a neuron. These patients had actually received BRDU for the treatment of other diseases. They passed away, and this group was able to obtain their brains and do these studies. For the first time, freezing these tissues in time, they were able to find evidence of new neurons being formed in the adult human hippocampus. So we ask ourselves the same questions. By 2004, we asked and published a paper in which we ask ourselves the questions: the adult human subventricular zone could potentially have neural stem cells. We actually obtained specimens from the operating room. We look at specifically specimens from the subventricular zone, and we were able to form these beautiful small spheres. We call them neurospheres. We purified this subventricular zone astrocytes. That means through a procedure in which we shake the cells, and at the end in the petri dish, we only leave astrocytes. And we were able to create even more neurospheres as compared to those episodes in which we did not purify the astrocytes. And when we obtained astrocytes from the cortex and the striatum, we had absolutely no evidence of sphere formation, telling us for the first time that indeed there's got to be something peculiar in the astrocytes that are found in the subventricular zone, because not only when we purify them we find more astrocytes and more neurospheres, but also when we have the same astrocytes or similar astrocytes from the cortex and striatum, we found no evidence of neurospheres. This is the picture of one of such neurospheres, and not only that, when we put them in a special media, we were able to form the three lineages, in this case neurons, in this case oligodendrocytes, as you can see right here, and astrocytes. So from one of these spheres, from the subventricular zone, we found the capability of these cells. This is a sphere that came from a single cell that was an astrocyte. It was a GFAP positive cell, and we differentiated the cells. We were able to produce three lineages, once again, astrocytes, neurons, and oligodendrocytes. But that wasn't enough. The question was, can human subventricular zone astrocytes produce neurons without the effect of growth factors. So what we did right here is we provide an environment in which we use astrocytes from the human brain, alrighty, and then we got one astrocyte from the subventricular zone and we dropped it in here to see whether or not, without putting exogenous growth factors, we could actually produce a neuron. And I'm inside of one of these experiments, and you can actually see in red a beautiful neuron that is being formed from an astrocyte that was derived from the subventricular zone without the use of growth factors. We try to do the same from cortical and striatal astrocytes, and we were unable to produce a neuron. Once again, telling us that there is something about the subventricular zone astrocyte that makes those cells peculiar and able to produce neurons in the actual human brain. Now, all this was done in the Petri dish, and yet we were able to find for the first time that in the region of the lateral ventricle, as you can actually see right here, there are several regions. This was actually after studying a lot of specimens, not only from the operating room, but also from post-mortem tissue. We were able to find that in the lateral ventricle of the human brain, there is a specific population of astrocytes that can give rise not only to those beautiful spheres, but also to neurons, oligodendrocytes and astrocytes, and most important, even without growth factors, we can actually produce a single neuron. So indeed, there is a possibility up until the present, what we didn't know before, that the human brain, at least in the Petri dish, has the ability to produce neurons. So there is probably a cell population that we call stem cells within this area of the subventricular zone. Other groups have continued to look on this cell population within the actual hippocampus, and they continue to move this field forward.